All right. Well, I think we'll get you started, Gabbie, because time is running on. My colleagues who are online now, you should know that we're really privileged to have Gabbie talk about this subject. Not only is the subject fascinating and probably pretty germane given the way we're seeing pandemics go these days, but she's actually in Paris. So would you believe it's about 3:33 in the morning in Paris? So I think this is a very brave and honorable thing that she's doing, giving us this presentation. So it's quite amazing. So Gabbie, as you see, is going to talk about avian malaria in Hawaii, and that's what she did her Ph.D. in 2020 at UC Davis with Tom Hahn, who we mostly know very well actually, as her advisor. So I think this is going to be fascinating, Gabbie. So I'm going to let you go ahead and do this. So thank you very, very much for the pleasure and privilege of you being with us. So over to you. Thank you.
Great. Well, Barry, thank you for that wonderful introduction. And thank you all. Barry and I were speaking these kind of few minutes before the presentation started. The internet may kind of come in and out from time to time, so there may be certain moments. Barry, certainly don't hesitate to interrupt me if there's a whole bit that kind of gets missed. But hopefully we'll cross our fingers that the internet behaves itself. Okay, so let's jump right into the material. That's obviously what you're here for. So today I'm going to be talking to you about a subject that is near and dear to my heart for many years now. I've been studying in some capacity the Hawaiian honeycreepers, a group of endemic songbirds to Hawaii that I'm going to be showing you some pictures of in a few slides for almost 12 years now. So it's a subject that I've become well acquainted with and that is important in conservation and in the realm of avian biology. So today. So I'm going to be talking about not only avian malaria in Hawaii, but also how the introduction of that disease has affected the conservation of the native birds in Hawaii and their evolution and what we can expect and kind of how we see this field evolving in the future. Okay. So before jumping into the specifics of avian malaria and talking about my study species for my dissertation, I want to talk a little bit about infectious disease in general. So this is a subject, as Barry alluded to, that has been on everyone's mind for the past. You all probably know, wild animals are affected by infectious diseases and it compromises their conservation in some cases. Now, birds are affected by various infectious diseases. We've got, they can be infected by ticks carrying Lyme disease, for example. Salmonella can also be a big issue in certain locations. This is a little clip that I took from a public service announcement talking about limiting salmonella outbreaks. And then, of course, there's the subject of my talk today, which is avian malaria. And that is a disease that is widespread, and most species do not die from the disease. There's not high mortality in most bird species. However, in certain island species, such as the honeycreepers of Hawaii that have evolved on Hawaii without lots of other blood diseases, are highly susceptible to diseases like avian malaria that have been recently introduced. So that's what I'm going to be talking a little bit about today. Now, first, I want to introduce you to the Hawaiian honeycreepers. You may be familiar with this group of birds. It's a group of birds, as I said, that's native to Hawaii. We've got just a smattering of them here on the slide. Originally, there were over 50 species and finch ancestor that got blown off course, probably flying from Asia. Now, unfortunately, as is the case for many island systems now, in recent years, 17 of those species are now extinct that I've given the little skull and crossbones on my little diagram here, and 24 are considered endangered. And those are the ones with the little red X on them. And so as you see, there are very few that remain that are neither extinct nor endangered. And this is because of a variety of factors. And we have the usual culprits, particularly on islands. Introduced mammals and reptiles often have a significant effect on native bird populations because they don't have the normal predator cues. They don't know that a rat could eat their eggs, or a mongoose could eat and attack them, or a snake is going to come up and predate their nests. So the introduction of, particularly on Hawaii, the mongooses have had a significant impact on the native bird populations there. And it just as a little anecdote, one of the ways that we captured birds when I was in Hawaii doing my field research is that we would put out a speaker and do playback, play songs of Amakihi singing, and then the birds would come in, expedite our captures. And at one of the sites, there were so many mongooses that we would put the speaker down and they would come right up to the speaker, try to figure out where it was, thinking it was like an injured bird on the ground. So these guys are really, they can look innocent, but these guys are pretty nasty. All right, now in addition to introduced predators, we've also had direct take have an impact on native Hawaiian honeycreeper populations. Even before the arrival of Europeans to Hawaii in the 1800s, you had the Hawaii royalty, the ali'i, standards. Here we had a cape that was made with native bird feathers. And those were made by, using thousands of birds and their feathers, because these guys are pretty small, little songbirds, even smaller than like sparrows. So thousands of those birds were necessary to make these types of capes and feather standards for traditional purposes. Another one of those usual culprits is habitat destruction. So of course, Honolulu and the skyscrapers is not ideal habitat for the native Hawaiian birds. And so that's affected their populations as well. And then finally, more recently introduced diseases have had a significant effect on bird populations in Hawaii. And so the one that I'm going to be focusing on today is avian malaria. So avian malaria, for those of you who are not as familiar with it as I am, is a intracellular plasmodium caused by intracellular plasmodium parasites. So I just want to acquaint you here, if we're looking at a microscope slide here on the left. And the red arrow is pointing to a red blood cell that is and then the green one is pointing to a normal red blood cell. So you see that in that infected one, we've got this big kind of lighter purple parasite that's in that cell. So that is actually the malaria parasite there. So it's very similar to human malaria, found in the blood, as I said, and obviously transmitted by mosquitoes as well. So you must have mosquitoes for this disease to be transmitted. And then it's, as I said before, it's a widespread disease. It's found in almost all avian taxonomic groups and on all continents except Antarctica, due to the fact that mosquitoes that as well as the plasmodium parasites themselves are cold intolerant. So they don't like cold, so not able to live down there, but everywhere else, they're able to find habitats where they can thrive. Now, for many years, avian malaria was not found, as I said, on many isolated island chains, including Hawaii. Now, the introduction of avian malaria to Hawaii took place in 2 steps. As I said, you need the mosquitoes to transmit the disease. So even if there were lots of birds carrying the disease that were introduced to Hawaii, that could not take off and affect the native birds until you've got that from one bird to another. So In the initial introduction that kind of led to the outbreak of avian malaria in Hawaii was the introduction of mosquitoes. I always think about how much of a real paradise Hawaii must have been mosquito-free, because, you know, today, for any of those of you who have visited or visited other tropical locations, you can pretty much assume that if you're in some sort of tropical forest, you're going to have mosquitoes. But before the 1800s, there were no mosquitoes found on Hawaii. Now we detect about 5 or 6 species, depending on which island you're on. And then initial introduction took place probably documented that whaling ships that were offloading barrels likely introduced mosquito larvae in offloading those barrels in Maui. And then after that, as you can expect, Hawaii is just prime habitat for mosquitoes. And so following that initial introduction, populations just went wild. The actual introduction of malaria didn't take place for a few dozen years later. It's expected that the main introductions that then led to the increase in avian malaria in Hawaii took place in about the early 1900s. At this time, because of other factors, the direct take and introduction of introduced predators that I was talking about before, those had already started to have a pretty significant effect on native populations of birds in Hawaii. And so folks were, particularly bird watchers in Hawaii, were kind of that they didn't have robust populations to watch and appreciate and enjoy, particularly at low elevations anymore. And so they thought, and as was kind of the general idea at the time, that, well, it would be really great if we introduced some exotic species from other locations here, and that way we'll have thriving bird populations. And they didn't at all have our knowledge and understanding of how the introduction of exotic species could then affect ecosystems at a much bigger scale. So species such as the Japanese white-eye on the top here, and even northern cardinals at the bottom here, were introduced to Hawaii during this early 1900s phase. And many of those were carrying avian malaria. So you've got our vector population that was introduced. Now we've got this nice pool of infected birds. And then after that, the disease just took off. And in the mid 1900s, but really by scientists had documented declines in the native birds as a result of the introduction of avian malaria. So one of the unique things about avian malaria in Hawaii is that we still have some high elevation refugia, they call them, where there is no avian malaria. So as I said before, no avian malaria in Antarctica because it's too cold there. Well, the same exists for high elevation habitats. So in Hawaii, you've got some mountains that get pretty high up. And at those high elevation habitats where you still have trees and plants can use as food and habitat. So it's not like completely, deserted and just a rock. So at those high elevation habitats, endemic birds and native birds of Hawaii are still able to thrive. Because there are no mosquitoes and thus no avian malaria. Unfortunately, at low elevation habitats, however, avian malaria is prevalent because of the robust mosquito populations there. So you've got this kind of interesting dynamic that you've got, particularly in kind of thinking about the evolution of species, is that you've got these populations that are exposed disease habitat at high elevation where you don't have avian malaria, and then also a lot of other diseases that have been introduced, avian pox and also avian mange, two other diseases that are transmitted both by mosquitoes, but also, well, the one avian pox by mosquitoes, but they can also be transmitted just bird to bird. Those are also a lot less common at high elevation. So you've got this kind of interesting thing going on. And so What's interesting is that, as I said, most of the endemic birds now species in my dissertation, the Amakihi, has been able to maintain stable populations, not only at high elevation, but also at low elevation. So this prompted scientists in about the 1990s, when they first discovered these stable populations at low elevations, they asked themselves, what is it about these birds that allows them to be able to cohabitate with this disease? What is it about them that is allowing them to survive? So there are kind of three different main ways that potentially the Amakihi was able to survive infection. First, potentially they're evading infection, some sort of behavior or other way of being able to repel mosquitoes and evade infection altogether. So that's one possibility. The next possibility is that they are potentially resistant to infection. So resistant meaning that they're able to limit that parasite load. So that's why I've got the little avian malaria in the circle and X through it. They're able to actually get rid of those malaria parasites. And then the third option is that they're potentially tolerant to the disease. They become infected, the disease is in them, but they're able to kind of mediate the negative effects of infection. At low levels, but are able to continue living and able to cope with that way. So these are kinds of the three main options that we have in front of us. And so we'll look at two different studies that help shed light on what may be going on in these low elevation Amakihi populations. So first, to determine whether they are evading infection. Well, if they're evading infection, then that would mean that low elevation populations are not infected with avian malaria, similarly to high elevation populations. So to determine this, they went out, collected blood samples from high elevation Amakihi and low elevation Amakihi. And then we're trying to determine, they used PCR to determine malaria infection status by looking for those genes of the malaria parasites in the birds. And so what they found, as expected, very few high elevation birds were infected with malaria. Now you do see that it wasn't 0, it was 1.5%. And this is likely because during the summer months in Hawaii, high elevations can host some kind of temporary mosquito populations that can then be transmitted. Birds can become infected even at high elevation over the summer months, and then they remain infected for life. So that explains that 1.5%, but essentially that is very close to 0. At low elevation, however, as you see here, they had 85% infection rates. So really most of those birds, almost 9 out of 10, were infected with avian malaria. And so clearly this is indicating that they are not evading infection at a very high rate. So we can cross evading infection off our list here. So now we want to consider the other two options. Are they resistant to infection? To limit that pathogen burden better than, are they able to do that? Or is it that they're just able to limit the health impact of the pathogen burden? That they become infected and they kind of limit that pathogen burden somewhat, but do kind of maintain a certain background level of infection. Now, this may seem like we're kind of splitting hairs here. Okay, if they become infected, you know, that's the most important thing, that to kind of then consider how that will affect their conservation and the long-term viability of the species. But this is actually a really important distinction to make in understanding the birds, because it's going the kind of coevolution of this disease with the bird is going to go in a very different direction if it's limiting the pathogen burden, getting rid of them altogether, or if it's maintaining some sort of low-level infection and allowing that disease to kind of affect the populations that way. So in order to try to figure out whether they're tolerant or resistant, this required an experimental study. So researchers over at the USGS, Carter Atkinson and his collaborators, organized an experiment where from high elevation and from low elevation. These were uninfected birds. They brought them into the lab, and then they experimentally infected them using mosquitoes that were carrying avian malaria, and then saw what happened. And so what they found was very interesting. They found that both high elevation and low elevation birds essentially developed the same parasite load during infection. You get this big ramp up of malaria parasites when they first become infected. It's called the acute phase of infection. So you get this big increase in parasite load. And both high and low elevation birds experience this really high increase. But the difference, the main difference, was not only that the high elevation birds experience much higher mortality, so the low elevation birds had lower mortality, but they also were able to maintain a more normal weight, and they were able to maintain normal feed more than the high elevation birds. One of the main signs of avian malaria in birds, particularly those that are highly susceptible to the disease, is they just stop eating. They stop moving, and essentially the disease just runs its course, and at some point they just run out of resources and aren't able to continue living. So these low elevation birds somehow are able to maintain a more normal weight and feeding that allows them to then get through that disease. So all of this then points to these low elevation birds being tolerant to avian malaria. So now that we kind of, this is all arrived and looked at these two main studies, the one in the field where they found the 1% at high elevation infected, the 85% at low elevation, and then this lab experiment. And so the question that I wanted to answer was, what are the mechanisms and consequences of this avian malaria tolerance in Hawaii Amakihi? So thinking about not only what is allowing them to tolerate this disease, to cope with infection in this way, and then also better coping with the disease if they have higher immunity as a result of coping with this disease. What does that mean for their survival, for their reproduction, and their long-term viability? All right, so I'm going to be asking, going to introduce two different questions then that I asked in the context of my dissertation research, and then give you some of, show you some of the data that I found, and also show you some neat pictures of Amakihi and other birds along the way. So the first question that I wanted to ask was, how do immunomodulatory hormones contribute to avian malaria tolerance in low elevation Amakihi? Now that's a mouthful, so let me walk you through this for a second. Immunomodulatory hormones, well, those are hormones that affect the functioning of the immune system. They modulate the immune system, either increasing it, stimulating it, or suppressing it. And so there are multiple hormones that do this. You may be familiar, a lot of popular press articles have discussed the potential role of testosterone in kind of mediating COVID infection, and kind of thinking about the differences in men and women and how they've reacted to the disease, and how potentially testosterone may be playing a role there because it can be an immunosuppressor. So the hormone that we're going to be looking at today is corticosterone. So that is very similar to cortisol, which is the stress hormone that's found in mammals and even humans. And this is a hormone that increases when you're stressed. So if someone comes up behind you and surprises you, your cortisol is going to go through the roof. If you're dealing with really stressful situations at work, kind of day after day, your cortisol is probably anticipating a stressful experience. Your cortisol is going to be high. And so the same happens in birds. If they're attacked by a predator, if they're experiencing a strong storm, their corticosterone is going to be high. Now, corticosterone is important because it also has many effects, not only on regulating the stress response, helping those birds out of those stressful situations by causing various physiological, morphological, and behavioral responses to that stressor. But it also has a variety of effects on immune function. So you can see that potentially corticosterone here could be playing a role in kind of thing that I wanted to see. If we go out in the field, are there differences in corticosterone levels in these high and low elevation birds? And could that potentially shed light on why we see these differences in tolerance across elevation? So first, I was initially going out into the field and predicting that potentially, because of the variety of immunosuppressive effects of corticosterone. So corticosterone suppresses the immune system in many contexts. As a result, I was predicting that in these low elevation birds, corticosterone would be lower. So lower corticosterone, lower suppression of the immune system. All right, so I'm gonna get you into the methods of my field research here. This is just a picture of me and my field assistants going out to one of our fields. No, I'm just kidding, obviously. Here are some actual pictures of me and my field assistants out in the field. So I just want to give a couple of shout outs here. This is one of my fantastic field assistants, Molly Heel, who was very brave one day when we caught a native Hawaiian hawk, an eo, in one of our nets. So I had never touched anything larger than a cardinal in my life. So I was very grateful that she was there to take care of this guy. So that is one of my field assistants there. Here, you've got a picture of me holding an Amakihi. They're really pretty small, as I said, and feeding with a pipette, feeding it some sugar water that we would just get off and go on their way. But sometimes it was good to give them a little bit of a pick-me-up before taking off, similarly to humans, to us getting some sugar juice, some juice or a cookie after donating blood. So that's what I'm doing there. And then another one of my field assistants here, Kathleen Hunt, who is taking a bird out of a mist net. You can't see it too well there, but we'll have another picture where you'll be able to see it better. And then a variety of pictures from some of my field sites here, just to kind of give you an idea. Some groups that I talk to have a very different vision of Hawaii than the one that I'm familiar with, which is the mountain Hawaiian up in the forest. Other folks are more kind of familiar with the beach side of Hawaii. So just wanted to give you an idea of what my day's work was like there. All right, so we went out into the field. Now this research I was conducting on Hawaii Island. It's where the Amakihi populations are most robust and also where they've been most studied. And so it was just kind of the best way to be able to build off the research that's been conducted in the past. So these are my field sites in the red circles here. So I had 3 high elevation sites. Those are the 3 there. I had sites both on the wet side of the island, which is on this side, and then the dry side indicated by the sun. And that was because mosquito populations can vary in size based on water availability, habitat availability as a result of higher or lower rainfall. So I wanted to kind of get field sites on both the dry and wet side of the island. Now here we're looking at an Amakihi that's been caught in a mist net. You may be familiar with this technique, but just as an overview, you put up these big nets that are essentially sometimes if there's high wind, they can see them, but essentially they're not visible to the birds. They fly into the net, fall into these pockets, and we pull them out of the net and then take our blood samples. So the corticosterone that we were measuring, you can measure it in a variety of ways. You can look at fecal corticosterone in feces. You can also look at it actually in feathers. But what we were interested in looking at was the levels in the blood. So we were taking small blood samples and then also took a variety of morphological measurements to associate with those corticosterone levels. All right, so I'm going to now walk you through we were getting and then explain how we then moved on to the next step. So first, I want to show you a couple of beautiful pictures of Amakihi that we had captured. We took data from both females. So we've got a female on the left there and then a male on the right. The males are distinguished by that really distinctive black mask that they have. And then their plumage is much more bright yellow. Females are more of a drab color, is often the case in the animal world. And then have a kind of duller gray mask. So took blood samples from both males and females. And so what I'm going to be presenting to you here is my data on the hormone corticosterone in these birds. And what you'll see, so we've got corticosterone along the y-axis there. So that's the level. Higher is higher of these stress hormones and lower is lower levels of these stress hormones. Then we've got time after capture along the x-axis. Now, the reason I have that is because not only were we interested in looking at their stress hormones at normal levels, so if I came and measured your stress hormones right now, that would kind of be, at normal levels, you're not very stressed, you're kind of sitting here watching a presentation. But if I came up and surprised you, or hormone levels, the stress hormone levels would increase. And that's another measure that we can look at in birds as well, using as the stressor. So instead of jumping in and surprising you, the bird is captured. And so that is a stressful experience. And so we can use that capture in order to then look at that increase in stress hormones that they experience. So not only look at their initial levels, their baseline levels, I'll call that, but also their stress-induced levels, how that they experience that increase as a result of the stressor. And that gives us a different kind of information. So that's what we're looking at here. So we've got our initial time point that we took at close to 0. We captured sample as soon as possible. And then we take additional samples 20 minutes after capture and then 60 minutes after capture. And so that'll give us this idea of that increase in cort over time. And so what we see, so let me orient you. I'll be using the same color scheme throughout the presentation for a couple of other graphs I'll be showing you. And so the low elevation data, the low elevation birds, that's going to be in blue. And then the high elevation data is going to be in brown. And this graph here, we've got dotted lines for the males and then solid lines for the females. So what we see, and what I want to call your attention to, particularly among the males, the two dotted lines that we have here, is although it's not very visible on the graph here, but levels were not different for by elevation for those initial baseline levels before they what we see is that when we get to the 20-minute time point and the 60-minute time point, when we look at that cort ramp up as a result of the stressor, the high elevation birds have significantly higher levels of cort than the low elevation birds. So they've got a different response. It's that they both are increasing cort as a result of the stressor, but those high elevation birds have a much higher response to the stressor than the low elevation birds. So based on these findings here, okay, we didn't see a difference when they're not stressed, but we did see this difference in response to a stressor. So the next question that I then wanted to can help to explain something about how the birds are tolerating avian malaria. So the specific question that I wanted to ask was, is there a cause and effect relationship between these CORT levels that we saw in the field and tolerance? So can we link the difference in stress response that we saw in the field to tolerance through potentially its effects on immunity, through potentially its effects directly on parasite load? So that is what I wanted to investigate next. Now, to look at cause and effect relationships, you're probably thinking, you can't do that in the field. You're going to have to manipulate something. And that's true. So this required us to set up an experiment in the lab. So just as a summary there, given that CORT can have suppressive effects on the immune system, it can decrease, weaken immune responses. What I was predicting is that if we increase experimentally the CORT levels in these birds, what we would expect to find, if there is a link between these differences in CORT that we saw in the field and tolerance, we would expect potentially lower immunity in those birds that have the higher CORT, and potentially higher parasite load too, that would then lead to higher parasite loads. So that's what I wanted to look at next. So as I said, to do this, this involved doing a cap. Just wanted to show you a few pictures of what that kind of involved. We weren't in some lab somewhere in lab coats. And so we've got in the middle at the top here, this is me setting up some cages where we've got each of the birds. We had one bird per cage, but there were multiple cages per room. And so the birds were able to communicate to each other. They weren't in isolation. And each of the birds had food. You can see it a little bit better in the bottom left-hand photo there, a little food dish. Also these tubes that had a special type of nectar that was enriched with both proteins. So it obviously has like sugars and stuff, but also was enriched with proteins and other micronutrients. So that is mostly what they were feeding on when they were in captivity, but also gave them a variety of solid foods that they could kind of pick at. There were certain birds, there was one in particular that we gave them a few their solid food, and one of them that always pulled them out and threw them on the ground. And every morning, like clockwork, he would come in and have those blueberries on the ground under his cage. So included a little bit of vegetation. They didn't seem to really care about that or not, but I'd been encouraged to include that, and hey, why not? And then, so this is just a view of the aviary from the outside. This is, you probably recognize Tom looking at some of the samples that we had taken. And then this is us transporting the top right picture. The small temporary holding cages that we were using to transport the birds, put one in Amakihi in each little slot there, and transported them that way. So, all right. So let's jump into kind of orient you to what this experiment involved, and then we'll look at a little bit more data. Okay, so at about 10 to 15 days before the experiment, we captured the birds and brought them into captivity. This allows them time to acclimate to captive conditions, because as you can imagine, being brought into captivity can be stressful. So this, in other birds, has been documented stress hormone levels. So we brought them into captivity to allow them to acclimate to those conditions a few days before the experiment. Then on day 0, we took an initial blood sample, and then we implanted each bird with either an implant that was filled, it's a small plastic implant that was filled either with corticosterone steroids, corticosterone hormones, or that was filled just with water. So we had our control that just had the implant filled with water. And then we had the experimental group that had the implants with corticosterone that then would be secreted and increase their And these implants, it's really a relatively uninvasive experiment. You are able to slip them under the bird skin, which is not like mammal skin. It's not vascularized. So you can we were able to cut just with scissors, a tiny hole, slip the implant in under the skin, close that up with a special type of adhesive, biological super glue, and then release the birds back into their cages. And all of them seem to deal with this procedure just fine. And just as an anecdote, I captured some birds after the experiment for something else. Gone through the experiment, which I don't know if that boasts much of their memory of having been in captivity and been captured and gone through this whole experience and then get captured again. But it reassured me that we were able to conduct this experiment, release them back into the wild after the fact, and that they thrived. So that was nice. Getting back to the experiment though, we then took additional blood samples. So we took that initial blood sample at day 0 to get an idea of what are their levels before any type of manipulation. The samples then at day 2 and day 4 were then to figure out, okay, based on these levels that we have experimentally elevated, what's happening for the immune system and for parasite load. So we took additional blood samples on days 2 and 4, and then we removed the implants using the same procedure as we put them in on day 4, held them in captivity for a few days later just to monitor their activity. And then we're able to release them all back to their sites of capture at the end of that period. All right, so let's look at a little bit of the data that we found from that. First of all, this is kind of just something that obviously I would have to present in article now, but I also just wanted to walk you through this because it's an important part of the experimental process that I wanted to familiarize you with. So here we're looking at the corticosterone levels. So yes, as we would expect, the implants, we would expect, okay, that's going to increase the cort levels. Obviously, we're giving them extra hormone, but it's an important step to demonstrate that what we were planning on intending to do did actually work, because potentially, giving the implants also increased their stress levels. But what we found was as we were hoping. So we're looking at, again, we've got and then the high elevation data in the blue. And so, sorry, the opposite. As I'm sure you all are looking at the legend there and you're like, nope, that was wrong. Low elevation, like the ocean, is blue. High elevation, like the mountain, is brown. So regardless, what we see here is that the important distinction to make is the birds with those cort implants are in the dotted lines, and the birds with the sham implants, those are the ones that were filled just with water, were the solid lines. And so we see the solid lines are way down here. On day 0, of course, everyone's the same. But then on days 2 and days 4, the birds with that extra cort had much higher cort levels. So as expected, the implants did what we wanted them to. So check. Got that done. So now we can get into the more interesting effects on immune function and parasite load. So we're going to be looking at data from 2 different immune experiments here. So the first one is called the Hemagglutination Experiment. All this means is that we were looking at natural antibodies. So as you know, when we're getting the COVID vaccine or other vaccines, our bodies produce antibodies against that specific disease. However, we're also all born with, and this applies to birds as well, natural antibodies. And so those are just antibodies that we're born with. So this experiment, allows me to quantify those. So this is just a picture of what this experiment involved, and don't need to get into the details, but essentially I was combining rabbit blood with the bird blood, and then looking at how well those natural antibodies were able to cause agglutination of those rabbit red blood cells. So what we're looking at here, and just as a reminder, remember we were expecting there to be lower immunity in those birds with the higher cort because of its suppressive properties. What we see, so agglutination titer, that's just a fancy way of saying the number of natural antibodies. So higher agglutination, higher concentration, more natural antibodies, and lower is lower natural antibodies. So what I would have expected was that the birds with the cord implants were going to have lower natural antibodies. It was going to suppress those natural antibodies. As you see with this graph, though, we've got a little bit of fluctuation over the experimental day. So again, we've got experiment day down here on the x-axis. So 0 before we did any manipulation, and then days 2 and day 4 after. We don't see any significant difference over the experimental period. And one of the reasons I wanted to show you this is that often we'll kind of curate presentations to talk about the data that's most interesting. But I did want to remind you as well that often we'll do some sort of experiment and doesn't turn out the way we wanted it to. But that also provides us with more information that Amakihi that are exposed to higher levels of cort, higher stress hormones, their natural antibodies are not affected. So that's also an interesting point to take away. So didn't find what we were expecting here. So let's move on to leukocyte counts. So leukocytes is just another word for white blood cells. And so what this involved is that we took a small amount of the blood that we had sampled. We we put it on a microscope slide, and then we looked at that blood under the microscope. And we quantified the number of white blood cells. So the picture on the top right, you can see recognize what you see mostly on the slide there are red blood cells. So you probably recognize that from when we were looking at the malaria parasites in those red blood cells. So most of these are just normal red blood cells going along. And then we have a number of, under the red letters A, B, and C, we've got 3 types of white blood cells. So what we did is that we looked at these many fields of these cells under the microscope and quantified the number of white blood cells. So again, here we would be expecting that those cort implants would decrease those white blood cells in the birds. Now, this graph looks a little bit different than the other ones that we've looked at before. We're looking at up by both the elevation as before, high in brown and low in blue, but then also split up by implant type. So the sham, again, are the water implants, nothing in them, and the cord implants are the ones filled with cort. And what we see is really rather interesting. So let's look at the sham data for a second over here. First of all, we see that in the birds that were not manipulated, low elevation birds have higher leukocytes per blood volume. That's what we have along the y-axis there. Essentially, it's just a way to normalize leukocytes across the individual. So higher amounts, higher numbers of white blood cells, lower amounts of cells. So that those low elevation birds have higher levels of white blood cells than the high elevation birds do. But what's interesting is that then when we look at the cort birds, so the birds that had the higher cort, what we see is the opposite of what we were expecting among the high elevation birds. So low elevation birds, as you can see, it's pretty much equivalent across the board, and there is no statistical difference between those groups. So no effect, as was the case with natural antibodies, of those cord implants on the low elevation birds' white blood cells. The high elevation birds, however, we see that the white blood cells were significantly higher in the cort compared to the sham birds, which is the opposite of what we were expecting. So interesting finding here. All right, and then finally, we're going to wrap up with some of the parasite load results, and then I just want to kind of step back with some conclusions, and then I'll be happy to take your questions. So bear with me with just one more graph here. Plus, it's not, I'm still struggling to figure out how best to display this data. So we're looking at, again, we've kind of returned to that, the way that we were looking at the data before. We've got experiment day along the x-axis. So 0 is before that initial sample, and then days 2 and day 4 are measuring the effects of the implants. And this, having white blood cells along the x-axis, we're looking at parasite load along the x-axis. So higher numbers of parasites at the higher numbers, and lower numbers of parasites in the blood. And so what we see, so this is actually looking at each specific bird. So each line represents a specific bird. And as you can see, so we've got 8 birds that I'm representing by this graph here. Now you don't see 8 lines because there are a bunch that are kind of collected all at the bottom here. But what I wanted to draw your attention to is the ones that are much higher up here, the ones that we see a change in over the experimental period. And we're looking at only 8 birds here. We involved 40 total in our experiment, but only 8 of those were infected with avian malaria. So those are the birds we're looking at here. The birds were in a avian malaria proof, mosquito-proof aviary. So once they got in, if they weren't infected at the beginning, they weren't going to get infected during the experiment. So that's why we're only looking at these 8 birds here. But what's interesting, although our sample sizes are pretty low, so we're going to have to do follow-up studies, but what we see is that those birds with the cord implants, so our dotted line birds, the 3 that had the highest increase in parasite load also had cord implants. And then the birds, we had 1 bird that had a water implant that increased just very slightly by the end of the experiment. But otherwise, the other bird that had the water implant didn't increase at all. So we see this interesting difference, and our statistical models helped us to determine that there was a statistical difference in parasite load by the end of the experiment, with those birds with the cord implants having higher parasite loads than the ones with the water implants. So it does seem as though these cord implants are somehow, through their effects on immunity or through their effects on other components of physiology, are affecting the parasite load in these birds. Now, how it's doing it, we don't know, because as I just had shown you, the effects, there were no significant effects on immune function as we measured it here. All right, so after all of that, I'd like to take you back to the 10,000-foot level, because it's all well and fine to look at these detailed results of analyses and whatnot, but unless we're able to put it into some sort of context and tell a story with them, it's difficult to kind of move forward from there. So I want to kind of go back to the reason that I was interested in conducting this research in the first place. The Hawaiian honeycreepers are native to Hawaii, found only in Hawaii, unique biodiversity that we can find in one biodiverse, rich place in the world. And there are many things that affect these populations. We went through a number of them: the introduced species, direct take, habitat destruction, and all of these different variables not only can affect their survival, obviously, but also have other effects. That's why I was interested in looking at a hormone like corticosterone, but also why we could be interested in looking at other types of hormones and other types of their physiology. So not only should we be interested in looking at, okay, is the bird able to survive or not, or reproduce or not, given these introduced predators or other variables, habitat destruction. But what I'm interested, what was interesting me here was looking at various variables, whether it be the increase in skyscrapers in various places in Hawaii and Honolulu, whether it be natural disasters, lava flow. I was fortunate enough to be able to witness the 2018 lava flow to some extent when I was there. Then there are also other things that can affect these bird stress levels. So not only human destruction, but also natural destruction. Also other introduced diseases. As I had mentioned, there are other diseases. This is a photo of a bird that was affected with avian mange. And then also, even just as by us conservationists and biologists, we can increase these birds' stress levels and affect their physiology. There have been a number of captive programs that have helped in reintroductions of birds to the wild, trying to reintroduce birds to other locations across Hawaii. And all of those processes, holding birds in captivity, transporting birds, those can all affect their physiology. And I hoped I've convinced you over the course of this presentation that then those changes in physiology can also have an effect on, to less of an extent as demonstrated by my data, but they can affect then other components of either their reproduction, their survival, their immune function, as well as their ability to cope with infection. And then clearly all of those variables then affect our ability to be able to manage and conserve these populations over long periods of time. So I think not only do we need to interest ourselves in how are these various variables are effects on ecosystems and populations, not only how are they affecting just purely if the birds are able to survive or not, but also how is it affecting their physiology and how could that then influence their survival, their reproduction, reproductive effort, and thus our ability to manage those populations properly. So with that, I just want to thank a number of people, both in the field, the lab, particularly Carter Atkinson, who had done that initial captive experiment and was just a huge help throughout my dissertation. Beth Schultz, Jesse Krause, who helped with some of the experiment analyses in the lab. Of course, my advisor, Tom Hahn, and my dissertation committee members, John Wingfield and Kurt Claesing. And then, of course, everyone else who's up here on the screen, but I just want to give those shout outs. So with that, and of course, just want to thank my funding sources as well. Without their support, this research would not have been possible.